Hi again. Here in lesson 2 on the muscular tissue, we will look further into the anatomy of each muscle cell. And at the microscopic level, we will look at the filaments and the proteins that interact to create contraction force. We will also look at the events of the neuromuscular junction, which couple excitation to contraction and put it all together to understand not only contraction, but also relaxation. This diagram, which shows a sarcomere and its components, is very important for understanding the appearance of muscle tissue as well as its physiology in the context of muscle contraction. So let's go through it. The filaments, thick and thin, of myofibrils are arranged in repeating functional units called sarcomeres. Let's look at the various components. Plates of protein called Z discs separate one sarcomere from the next. So sarcomere is a space or the region from one Z disc to the next. Within a sarcomere, the thin filaments are attached to the Z discs and extend towards the center of the sarcomere from either side. So the D discs help to anchor the thin filaments. The center of each sarcomere is where the thick filaments are located. The thick filaments are composed of myosin protein, which are anchored by supporting proteins that make up the visible line at the center of each sarcomere called the M line. Now, the area spanned by the length of the thick filaments is called the A band. At the end of each A band is a zone of overlap, where the thin filaments overlap with the thick filaments. At the center of each A band is a slightly lighter region containing only thick filaments and no thin filaments. This is called the H zone. So at the center of the A band, you have the H zone, the naked thick filament zone, if you will. And at the center of the H zone, we have the M line, which is anchoring these thick filaments. Beyond the A band on either side are areas that contain only thin filaments with no overlap with the thick filaments. This region is called the I band, and it extends across adjacent sarcomeres and is intersected by a Z disc. This arrangement of filaments and other proteins within myofibrils creates the visible striations of skeletal muscle tissue, where the I bands create the lighter areas and the A bands create the darker areas. This arrangement of overlap between thick and thick filaments is also responsible for the generation of force and the shortening of muscles as they contract in a mechanism now designated the sliding filament mechanism. All right, so did you get all that? So that's the microscopic histological anatomy of skeletal muscle tissue. Again, the A bands are the darker middle parts of each sarcomere. They contain areas of thick and thin filaments that overlap at the ends. They also contain the H zone, which is the center of the A band, where there's only thick filaments but no thin filaments. And at the center of the A band, in the middle of the H zone, is the M line that supports the thick filament proteins and holds them in place. The I bands are the lighter bands, and these contain thin filaments but no thick filaments. I bands are formed by the ends of adjacent sarcomeres and therefore are intersected by Z discs. The Z discs are the bands of proteins that separate one sarcomere from the next and also anchor the thin filaments of the I bands. So this table summarizes the components of the sarcomere and also provides a lovely micrograph on the right, where you have your A bands creating the darker regions within the myofibrils and the adjacent I bands created by the thin filaments. And if you kind of squint, hopefully you can see the repeating light and dark bands that create the striations. The contractile organelles, the myofibrils, are built from three kinds of muscle proteins. First, we have the stars of the show, the contractile proteins. These generate force during contraction as they interact with each other. The contractile proteins are myosin, found in the thick filaments, and actin, found in the thin filaments. Next, we have the regulatory proteins. These switch the contraction process on and off by limiting or allowing the contractile proteins to interact. Two distinct regulatory proteins are found in the thin filaments associated with the actin. These are troponin and tropomyosin. Finally, we have the structural proteins. These work to align the thick and thin filaments properly. So we saw the Z disc proteins for the thin filaments and the M proteins that align the thick filaments. We also have proteins that provide elasticity and extensibility, such as the titin proteins found associated with the thick filaments. We also have proteins like dystrophin that link the myofibrils to the sarcolemma and help to transmit the force of contraction to the membrane, to the connective tissue layers, to the tendons, to the bones. So here we have an overview and categorization of these various muscle proteins. Again, myosin and actin are the contractile proteins, myosin in the thick filaments, actin in the thin filaments. The actin is surrounded by the regulatory proteins, troponin and tropomyosin, which essentially block the ability of myosin to attach to and pull on the actin to generate contraction force in relaxed muscles. When the muscle is stimulated, the tropomyosin is pulled away from the binding sites, allowing myosin to attach to actin in the thin filaments and pull on them, creating the motion that leads to force and contraction. Then we have several structural proteins. We mentioned titin, which connects the Z discs to the M lines of a sarcomere, thereby helping to stabilize the thick filament position. They have the ability to stretch and then can spring back unharmed, and therefore account for much of the elasticity and extensibility of myofibrils. Nebulin is a structural protein that wraps around the entire length of the thin filaments and helps to anchor them to the Z discs. And we have myomycin, a structural protein that forms the M line. And we have dystrophin. You might have heard of a genetic disease called muscular dystrophy. This is a defect in this dystrophin protein. This protein normally links the thin filaments of the sarcomere to proteins in the sarcolemma, and again helps to transmit the force of contraction to the sarcolemma. So let's meet these contractile proteins. Myosin is a protein that makes up the thick filament. It consists of a tail and two myosin heads, and the heads are the regions of this protein that bind to sites on actin molecules in the thin filaments during muscle contraction. The myosin also have binding sites for ATP. When ATP binds to the myosin, the energy of ATP is used to power contraction. Actin is found in the thin filaments. Actin proteins are more globular and are linked together within the thin filaments. Tropomyosin is a regulatory protein that wraps around the actin and protects or blocks the binding sites for the myosin in relaxed muscle. Attached to the tropomyosin is the protein troponin. Troponin binds calcium, which is released into the sarcolemma when muscles are signaled to contract. The binding of the calcium to troponin moves tropomyosin away from the myosin binding sites, allowing myosin to attach to actin and pull on it, generating the force of contraction. So calcium plays a critical role in the regulation of contraction. Without calcium being released from the sarcoplasmic reticulum when the muscle is signaled, the tropomyosin will block the binding sites on actin, preventing myosin from interacting and thereby preventing contraction. So here we have diagrams of the myosin made up again of a tail and two myosin heads. The myosin heads bind to specific sites on the actin proteins in the thin filaments. Again, the actin is covered by tropomyosin protein strands in a relaxed muscle. When signaled to contract, calcium that is released from the sarcoplasmic reticulum into the sarcoplasm binds to troponin. That binding changes the position of the troponin, and as it moves, it pulls the tropomyosin to which it is attached. Pulling the tropomyosin away now reveals the binding sites on actin for myosin, and now contraction may begin. We'll look at that in the next lesson.
All right, so now we're going to relate the anatomy to the physiology of contraction. As we consider the contraction cycle, it really begins with an electrical impulse from that somatic motor neuron. That then leads to the release of calcium, which is stored within the sarcoplasmic reticulum within muscle cells. As the calcium enters the cytoplasm, it then binds to troponin on the actin. And as it pulls the tropomyosin away from the actin, it reveals the myosin binding sites. Myosin then binds to the actin and pulls the thin filaments towards the center of the sarcomere from either side. This means that the thick and thin filaments slide over each other, and the thin filaments increase their overlap with the thick filaments as they're pulled towards the center of the sarcomere. With a strong contraction, the thin filaments from either side of the sarcomere can even overlap. This results in shortening of the muscle and is responsible for our understanding of how muscles generate force and contract. This is now called the sliding filament mechanism. So initially, scientists believed that the protein fibers were shortening themselves or coiling up on themselves, but upon visualization under a microscope, they observed that the length of the individual filaments did not change, but the overlap between them did. So this is how we now understand that muscles contract.
Let's take a look at what this looks like under a microscope. So, in a relaxed muscle, there is a slight overlap between the thin and thick filaments. The H zone at the center of the sarcomere, which again is a region that contains only thick filaments and no overlap, is relatively wide, and the Z lines are apart. As the muscle begins to contract, the thin filaments are pulled toward the center of the sarcomere into that H zone. This results in increased areas of overlap at the ends of the A bands, less areas without overlap, so the H zone narrows, and you'll see that the Z lines are coming closer together as the thin filaments which are attached to them are pulled toward the center of the sarcomere. However, the width of the A band does not change in size because those are the thick filaments. They are not actually moving. They are pulling the thin filaments towards the center of the sarcomere. The I band, on the other hand, is narrowing as the thin filaments increase their overlap with the thick filaments. And if you look at this final image in a fully or maximally contracted muscle, you'll see that there is maximum overlap between the thin filaments and the thick filaments that make up the A band. The H zone has essentially disappeared, as has the I band, while the width of the A band has not changed, and the Z lines are as close together as possible. This is reversed as the muscle relaxes. So as the muscle contracts, let's summarize the changes. The zones of overlap increase. the H zone narrows, the I band narrows, the A band does not change its width, and the Z lines come together. Let's now look at the events of the contraction cycle. So once the binding sites on actin are exposed, with the binding of calcium again to troponin, which pulls away the tropomyosin, and myosin is now allowed to bind to and interact with the actin, the repeating sequence of events that cause the filaments to slide begin, and these constitute the contraction cycle. There are 4 steps. Now it's a cycle, so technically you could start anywhere, but let's look at this first step, ATP hydrolysis. An ATP molecule that was previously bound to the myosin heads will undergo hydrolysis. This splits the ATP into ADP and phosphate, a phosphate group. The energy that is released reorients and energizes the myosin head. It gets it ready to bind and pull on the actin. In step 2, that energized myosin head now attaches to the myosin binding site on actin and forms what's called a cross bridge. So the myosin now is going to attach to the actin. The energy for this, or the change in the configuration of the myosin, occurs when the myosin head releases the phosphate group. So the release of the phosphate group, which was created in the previous step from the myosin head, is what changes the configuration of the energized myosin head. It now attaches to the actin and forms a cross bridge. Guess what's next? Pull! It's called the power stroke. So during the power stroke, the cross bridge rotates where the myosin head again changes its position and it pulls on the actin in the thin filaments, sliding it toward the center of the sarcomere. The change in the shape and position of the myosin that creates the power stroke is due to the release of the ADP, adenosine diphosphate molecule that was still attached to the myosin head. So by the end of the power stroke, the myosin head has nothing attached to it, but it is still attached to the actin. Finally, a new molecule of ATP binds to the myosin head. This again changes the configuration of the myosin protein, and the myosin detaches from the actin. Contraction involves a repeating of these steps so that the thin filaments are repeatedly pulled towards the center of the sarcomere during any one contraction phase. So it's like a hand over hand pulling of a rope towards you, or think of rowing a boat and progressively moving forward with each stroke. All right. Now, the binding of that new ATP is definitely responsible for detachment and the repeating of the cycle, but this also is responsible for relaxation. Muscles only relax when the myosin is dissociated from the actin. So ATP not only powers contraction, but ATP is also necessary for muscle relaxation. Also note that it is a fresh molecule of ATP that binds to the myosin head with each cycle, and this is why muscle tissue requires high amounts of ATP production and therefore has a high demand for oxygen, glucose, and other nutrients. Here's a depiction of the events we described on the previous slide. So let's start on the left of the diagram where there was a relaxed muscle and the myosin heads have a molecule of ATP attached. The ATP is hydrolyzed and split into ADP and phosphate that reorients the myosin head and energizes it. So that's step 1. In step 2, the myosin heads release the phosphate that changes the orientation of the myosin heads, and now they cock and attach to the revealed binding sites on the actin in the thin filaments, forming the cross bridge. In step 3, the ADP is released, and that causes the myosin head to rotate towards the center of the sarcomere, pulling the thin filaments towards the center. That is called the power stroke. It shortens the sarcomere if the resistance to shortening is overcome. And in order for this cycle to be repeated, a new molecule of ATP binds to the myosin head, allowing the myosin heads to detach from the actin and grab on again, repeat the cycle, and slide the filaments further towards the center of the sarcomere. All right, let's put some of these concepts together. We're almost there. All right, excitation contraction coupling. Let's look at how the electrical impulses that initiate contraction lead to the events of the contraction cycle. In a relaxed muscle, which is depicted on the left, the calcium inside the muscle cells is sequestered or stored within the sarcoplasmic reticulum, and the channels for calcium are closed. So there's no free calcium in the sarcoplasm. In the relaxed muscle, the thin and thick filaments of the myofibrils are overlapped, but the myosin is not attached to the actin. And this is because the tropomyosin is covering the binding sites for myosin on the actin. On the right side of the diagram, this is now a signaled skeletal muscle where the somatic motor neuron released its neurotransmitter and it led to an action potential along the sarcolemma that travels down the T tubules. As the signals reach the sarcoplasmic reticulum, it leads to opening of the calcium channels, and now calcium can diffuse out of the SR into the sarcoplasm. The calcium will bind to the troponin, pull away the tropomyosin, and this now reveals the myosin binding sites on the actin. Now, myosin and actin can do their thing. So this is now where we have the events of the contraction cycle that lead to the sliding filament mechanism. The myosin bind to the actin, pull them towards the center of the sarcomere, and will release the actin with the binding of a new ATP molecule, and the cycle will repeat as long as calcium is free in the sarcoplasm. So keep that in mind as we think about what is necessary for muscle relaxation. The extent to which a muscle can generate force is dependent on the length-tension relationship, and this is dependent on the length of the sarcomeres prior to contraction. So there's a sweet spot. There's an optimal amount of overlap between the thick and thin filaments that will allow for maximum overlap during contraction and therefore maximum contraction strength or force. However, when a muscle fiber is overstretched and there is too little overlap initially between the myosin and the actin, essentially the myosin heads cannot grab onto the actin, and this decreases the forcefulness of contraction. On the other end of the scale, if the muscle fiber is already shortened or compressed, then fewer myosin heads make contact with the thin filament and tension is also diminished. Let's look at it on the next slide. It should be clearer. All right, so here we have varying degrees of arresting sarcomere length. The tension is optimal when there is slight overlap between the thin and thick filaments. That's in the middle of the graph. Towards the left of the graph, we have an understretched fiber. This is one that's already shortened. There is already quite a bit of overlap and really not much more pulling that the myosin can do on the thin filaments to further shorten the sarcomere. Therefore, the tension or the force of contraction is diminished. On the opposite side of the scale, or at the right of the graph, we have an overstretched muscle fiber where the thick and thin filaments are minimally or maybe not even overlapping. This leaves little opportunity for the myosin heads to grab onto the actin and form cross bridges. Therefore, the tension drops significantly. Let's now move on to the concept of the neuromuscular junction. So as the name implies, this is the area within the muscle cells where the axon terminals from your somatic motor neuron extend to and lie across from the sarcolemma of our muscle cells. Action potentials arise along the somatic motor neuron and then lead to signal transmission to the sarcolemma of the muscle fiber. The synapse is where communication occurs between the neuron and the muscle fiber, and it involves a gap or a cleft that separates the two cells. Across the gap will travel the chemical called a neurotransmitter. A neurotransmitter is a chemical released from synaptic vesicles at the ends of the axon terminals or the synaptic end bulbs, and the neurotransmitter released from somatic motor neurons is acetylcholine, ACH. So neurons communicate with other neurons, or in this case, target cells, the muscle cell, by releasing a neurotransmitter that crosses the synaptic cleft and binds to receptors on the surface of the target cell. So the motor end plate is defined as the region of the muscle cell membrane, or the region of the sarcolemma, that is directly opposite to the synaptic end bulbs of the axon terminals. This is where the receptors for the neurotransmitter are located. So as neurotransmitter, acetylcholine, is released from the synaptic end bulb vesicles, the neurotransmitter crosses or diffuses across the synaptic cleft and binds to receptors for acetylcholine on the motor end plate, which is the region of the sarcolemma that forms the synapse at the neuromuscular junction. Hopefully, this becomes clear when we see it in a diagram on the next slide. In this diagram, we have the structures associated with the neuromuscular junction. On the left, we have an axon collateral of the somatic motor neuron. So this is a branch of the axon that further branches into axon terminals, and the axon terminals widen into synaptic end bulbs that store neurotransmitter in synaptic vesicles. That is enlarged at the top right. All right, so there we have the synaptic end bulb with the synaptic vesicles containing acetylcholine. When an electrical impulse travels along the axon and reaches the terminals, it stimulates the movement of the vesicles to the membrane where they fuse and release the neurotransmitter. Essentially, it's exocytosis. The neurotransmitter acetylcholine enters the synaptic cleft and then diffuses across that tiny space to bind to receptors in the sarcolemma of the muscle cell at the motor end plate. So this is depicted in the bottom diagram. All right, the binding of acetylcholine to the motor end plate receptors results in the opening of sodium channels now in the sarcolemma. The influx of sodium, remember sodium always diffuses into cells through sodium channels. The inward diffusion of sodium now initiates a muscle action potential that travels the length of the sarcolemma and down the T tubules and this will lead to the opening of calcium channels within the muscle cells located in the sarcoplasmic reticulum membrane. And you know the rest of the story, the coupling of this excitation event to contraction. Also shown in this diagram are enzymes called acetylcholinesterase that break down acetylcholine in the cleft. That is important for limiting the extent of contraction and for ending a contraction. As long as a somatic motor neuron is experiencing impulses and causing the release of acetylcholine, contraction will occur. Or as long as acetylcholine is in the synaptic cleft and binding to its receptors in the motor end plate, contraction will occur. So to end contractions and to control contractions, acetylcholine in the cleft has to be removed. One way it is removed is by breakdown by acetylcholine esterase, which is pre-existent in the synaptic cleft. The other ways that acetylcholine is removed is by diffusion out of the cleft. So it simply diffuses away and doesn't bind to the receptors. Thirdly, there are ATP pumps in the synaptic end bulb that actually pump acetylcholine back into the synaptic end bulbs. All three of those are required or contribute to relaxation of the muscle cell by removing acetylcholine from the cleft so it no longer signals a muscle action potential in the muscle cell that leads to contraction. So these events are further described here, occurring at the neuromuscular junction. As an action potential travels the length of an axon and reaches the terminals, it opens voltage-gated calcium channels in the synaptic end bulb, and this leads to an influx of calcium. So calcium concentration is higher outside of cells. When channels for calcium open, calcium moves into the cells. This is one of the major roles of calcium that we discussed earlier in the skeletal system. The influx of calcium is necessary for the release of neurotransmitter into the synaptic cleft, as the influx of calcium signals exocytosis of the neurotransmitter from the synaptic vesicles. We saw that acetylcholine will then diffuse across the cleft and bind to ligand-gated sodium channels on the motor end plate of the sarcolemma. That then causes an influx of calcium into the muscle cell. That changes the voltage in the membrane. It's called depolarization. And that depolarization initiates an action potential that travels down the T-tubules to the sarcoplasmic reticulum, leading to the opening now of intracellular calcium channels in the membrane of the sarcoplasmic reticulum. So this is distinct from the calcium that was outside of the neuron that flowed in to lead to neurotransmitter release. We are now talking about calcium that was sequestered inside the muscle cell within the sarcoplasmic reticulum. A muscle action potential will cause channels for that calcium to open. Calcium now diffuses inside the cell into the sarcoplasm, and this leads to the whole story about troponin, tropomyosin, and actin, cross-bridge formation, etc., etc. As we mentioned, in order for contractions to end, the neurotransmitter acetylcholine has to be removed from the cleft. It is broken down by the enzyme acetylcholinesterase. It also diffuses out of the cleft or experiences neurotransmitter reuptake as it is pumped back into the motor neuron. As long as the somatic motor neuron is firing or experiencing action potentials, acetylcholine will be released and will lead to contraction in the muscle cell. So the end of neurotransmitter release, or the end of signaling, as well as the removal of acetylcholine from the cleft, is required for relaxation. This is the super slide for this chapter. It puts everything together for us. Let's begin with excitation. at the top left of the diagram. So excitation involves a nerve impulse or an action potential that is initiated and travels along the length of the axon of a somatic motor neuron. As it reaches the axon terminals, the influx of calcium will then trigger the release of acetylcholine from synaptic vesicles. Step 2. Acetylcholine diffuses across the synaptic cleft, binds to its receptors in the motor end plate in the sarcolemma of the muscle cell. That then will trigger the influx of sodium that leads to a muscle action potential along the sarcolemma and down into the T tubules. Pause. In step 3, it mentions that the enzyme acetylcholinesterase in the synaptic cleft will destroy acetylcholine, so another muscle action potential does not arise unless more ACH is released from a motor neuron. Again, contraction will only occur if the motor neuron is signaling contraction and the release of more acetylcholine. Let's resume the whole contraction story. In step 4, the muscle action potential that travels down the T tubules results in the opening of calcium channels in the sarcoplasmic reticulum within the muscle cell. Calcium then diffuses out into the sarcoplasm. In step 5, calcium binds to troponin on the thin filaments. This pulls away the tropomyosin and exposes the binding sites on the actin for myosin. In step 6, we have the events of the contraction cycle that lead to the sliding filament mechanism. So that involves ATP hydrolysis and energizing of the myosin head, cross bridge formation, power stroke, as well as detachment with the binding of a new ATP molecule. And that cycle will continue as long as the calcium is still in the sarcoplasm and as long as there is available ATP to allow for detachment and re-energizing of the myosin head. As we move to the bottom left of the diagram in step 7, calcium release channels in the sarcoplasmic reticulum will close if there is no signal for contraction. So once acetylcholine is removed from the cleft, the channels close and calcium pumps in the sarcoplasmic reticulum will use ATP to restore low levels of calcium in the sarcoplasm by pumping it from the sarcoplasm back into the sarcoplasmic reticulum. So these calcium transport pumps are always active, always removing calcium from the SR. However, the calcium channels that allow calcium to diffuse into the sarcoplasmic reticulum remain open as long as there's a muscle action potential being signaled by acetylcholine in the synaptic cleft. Once calcium is sequestered back into the sarcoplasmic reticulum and is no longer in the sarcoplasm, tropomyosin will resume its resting position and will cover the binding sites on the actin. And now the muscle relaxes. So to summarize, in order for muscles to relax, there has to be a reversal or a cessation of all the events that led to muscle contraction. Number 1, relaxation requires no signals along a somatic motor neuron. Number 2, no acetylcholine in the synaptic cleft. Number 3, no influx of sodium into the sarcolemma, leading to motor action potentials. Number 4, calcium must be stored within the sarcoplasmic reticulum. That requires ATP. Number 5, ATP must also be available to cause myosin detachment from the actin. All of these are required for muscle relaxation.